Okay, so watch and listen carefully here, guys. Who was the first person to view cells? Andy. Hooke, right, okay. So, Hooke was the first person to view cells. 1 mark for Hooke. All right, the main reason for the differences between plant and animal cells is modes of nutrition. Okay, they get their glucose in a different way. After that, they're pretty much identical. Okay, how we process glucose and how a plant processes glucose is the same, chemically the same. Yeah, all right. Don't really like that, but okay. No, that's one of the differences, but it's not the main reason for the differences. Yeah. Okay, list the 3 points of the cell theory. So, first point is, right, all living things are all organisms made of cells. Okay, second point, cells carry out the basic functions of the organism. Third point, right, cells come from the division of existing cells or something like that. It's not that they're pumped out in a factory or created in any way. So if they mention something about cells reproducing by division, that's essentially what they need to have there. Okay, 1 mark for each of the 3 points. Okay. Number 4, when using a microscope, we always start with which lens? Scanning power. Number 5, what do we call an organism that must consume other organisms as a source of energy, right? A chemoheterotroph. Okay, and number 6, what's the main reason that we believe plants and animals have a common ancestor? Right, if they mentioned anything about structural similarities, procedural similarities, something about similarities, okay, then that's good. No, I wanted a specific name. Yeah. Okay, any questions for number 6? Okay, if they mentioned anything about the number of similarities, that's essentially the reason. Okay, number 7, a blank helps a plant cell to make sugar, while a blank helps an animal cell break down its food for sugar. Yeah. Chloroplast and lysosome, right. Okay, test is out of 10. Quiz, sorry, is out of 10. Please give them a mark out of 10 and let them see it. 1 second. Okay, obviously we didn't quite finish the plant cell, but I mean, the rest of the stuff we hadn't finished was all the same as the animal cell anyway, so it wouldn't be hard for you to just flip back and forth and figure them out. Okay, what we got to go over today is what do all the parts of that cell do, and how does the cell transport materials? Okay, and by transport materials, I don't just mean within itself, I mean how do they get in and out, all right? Because all cells have this membrane around the outside, and the membrane, like we said yesterday, is like border security. It's making sure that good things get in, bad things stay out, vice versa, bad things go out, good things come in, okay? But we're looking for how does that work, and how does it make sure that it gets things in that need to get in? Because the membrane is a barrier, okay? And some things can actually pass right through it, almost between the pieces of the membrane, but other things are too big, and they have to be packaged and processed and things like that in order to be brought into the cell. So we got to look at the different methods of cellular transport. That's what we're looking at today. Parts of the cell, what they do, and all the methods of cellular transport. Okay, so first point, we're going to understand the significance of compartmentalization of cells. Okay, compartmentalization means to break things up into separate compartments. And obviously, looking at a diagram of a cell, we see that there are lots of little compartments within the cell. Okay, that is true of eukaryotic cells, cells like ours that have organelles, nucleus, and all that kind of stuff. Okay, bacteria and things like that are a different kind of cell. They're what we call prokaryotic. They're from a more ancient line of life. They don't have all those organelles. Okay, so they function differently, and there's some advantages and some disadvantages for both. Okay, like I said, we need to know the role of each organelle. We need to understand that cells are a complex system of interdependent processes. Okay, what does interdependent mean? They rely on each other. If one thing goes wrong, everything else, there'll be like a trickle-down catastrophic effect. Okay, so they're interdependent on each other. One thing needs the raw materials from one other thing and so on. Okay, understand how the cell carries out its functions, and understand how materials are transported across the cell membrane and within the cell. Okay, so cells are divided into compartments. So we got all these little organelles. Each organelle has its own membrane. Okay, for the most part, the membranes of all the organelles are the same as the cell membrane. They're made out of the same stuff. Some organelles have thicker membranes than others. The nucleus has a special membrane that's a little thicker. The mitochondria has a special membrane that's actually a double membrane, and we'll talk about that in a little bit. Didn't I get a question? Breaking up into little rooms. Okay, so we've got these membranes that are separating one thing from another. Okay, the reason we want that is because sometimes the job that's getting done in one organelle would interfere with the job getting done in another. Right? So they operate best in their own little environment. Okay, and in that environment, they can have the conditions that they want in order for that thing to work most efficiently. They don't have to share space. Right? It would be like if you were trying to do math homework in the band room while the band was practicing, and there was a basketball game being played at the same time. Okay, now are those three things going to operate well in the band room altogether? No. Okay, people are going to get hurt. Okay, because the basketball is going to be flying around and people are going to be running in and out of the instruments. Okay, drum skins will be ruptured and horns will get bent and all that kind of stuff. Okay, and meanwhile you're sitting there trying to do your math homework and you can't concentrate because you're dodging basketballs and people are banging on instruments and blowing into them and making lots of noise. Okay, the three things just don't work well in the same environment. Okay, so we have within the school separate rooms for all of them because that makes sense. Okay, it's way easier to do math in a classroom where it's quiet and you don't have to worry about stuff hitting you, okay, or really loud noises going on all the time. Okay, that sort of makes sense. That's compartmentalization. Everything needs its own separate environment. That means they happen and they work most efficiently. Okay, and that's what we call, or that's what's present in eukaryotic cells. Okay, the other kind of cells are prokaryotic cells. And actually, we're going to have a whole, for AP anyway, have a whole separate lesson on eukaryotic and prokaryotic cells. Prokaryotic cells don't have organelles. So they are the band room with math and phys ed going on at the same time. Okay, all of that stuff has to happen in the same space, which is why prokaryotes typically do not get very big. Okay, why there are no multicellular prokaryotic organisms. They have no ability to specialize, okay, and do things like that. So they're much more, like we said, kind of from a more ancient line and a more simple line of life. All right, the other example here would be like running your furnace to heat your home, but running your refrigerator to keep food cold. Like basically you have three separate environments in your house: the freezer, the fridge, and where you live. All right, if you and basically that they each do their own job very well. You're comfortable at about 20 degrees. Your food doesn't spoil but doesn't freeze at about 4 degrees, and your food doesn't spoil because it's frozen in the freezer at minus 15 degrees. If you tried to do all of that in one environment, you would not be successful. Okay, because none of the three jobs would be done well. It would be, you'd probably have to keep your house at like 8 degrees. Okay, so you're going to be cold, your food's going to spoil, and anything that was in the freezer is certainly going to spoil, so none of the things are getting done very well. Okay, trying to find that happy medium just doesn't work. It's like trying to please everybody. Okay. All right, questions on that? Okay, so eukaryotic cells have this compartmentalization. So plant cells, animal cells, fungal cells, okay, protists, so the amoeba and the paramecium and all of that, they're all eukaryotic because they have all of these organelles inside. Okay. So the compartmentalization of cells allowed cells to become larger. Prokaryotes are usually pretty small, so this picture kind of gives you an idea of perspective. This is a typical animal cell. We don't even see the whole thing in the picture, okay? Because eukaryotic cells, they have a size range anywhere from, let's say, about 80 micrometers to even upwards of, let's say, 600 micrometers for a really big cell, okay? But a bacterium is quite a bit smaller. Okay, this is a prokaryotic cell, a bacteria. You can see inside this is the DNA here. It's not contained within a nucleus. It's just there, and so is everything else, right? Which is not really a good situation. But you can see it's only about a micrometer, maybe 2, okay, in size. And a virus is less than half of a micrometer. Okay, so very, very small. Because you don't have that specialization, you just don't get very big because things don't work very efficiently. It would be like if you didn't eat properly or didn't eat very much, didn't get enough calories every day, your growth would most likely be stunted, okay, as a result. All right, so allows them to become larger, and it also allows them to become specialized. That's a word that we're going to talk about more when we start talking about multicellular organisms. But specialized means within your body, your cells are not all the same. Muscle cells are different from nerve cells, which are different from the cells of your small intestine, which are different from your skin cells. They're all specialized to do a certain job, which means they may have more or less of a particular organelle. They may have a slightly different membrane. They may have the ability to make something that other cells don't have the ability to make. Okay, stuff like that. Questions there? All right, DNA. Deoxyribonucleic acid. We'll talk a little bit about that for AP as well. It's one of our extra lessons. Okay, in the environment of the nucleus, DNA in the form of chromatin is stored. So chromatin is basically the long, stringy strand, double-stranded helical molecule that you kind of associate with DNA. Okay, it's all stored in there in almost like if you had a ball of yarn and you completely unraveled it and then threw it in a pile. That's what the DNA inside your nucleus looks like. It is not really organized in any fashion, believe it or not. Okay, it's a library, but it's a library with no shelves. The books are just strewn everywhere. Okay, no Dewey Decimal System or anything like that. Okay, it's just everywhere. Luckily, your nucleus kind of knows where to find everything because it's the person that made the mess. All right. So that's what goes on within the nucleus. Is we got that special area where our most valuable resource for the cell is kept, okay, where the information for doing everything is kept. Now, obviously, you can see here that we have a special membrane, okay, and on that membrane are little pores, okay, called nuclear pores, and they're just a little hole that allows the copied DNA to get out. Okay, we talked about yesterday how when you need to make something, your nucleolus could just copy a section of the DNA and send it out. We'll talk a little bit more about that today, but that's how it works. It gets out through those pores and goes to the ribosomes, and the ribosomes put the proteins together. Okay, but it's basically a one-way street. Okay, very few things actually get into the nucleus. Most everything is coming out, and it's usually just copies of DNA. Right, so that's what the nuclear pores are for. So it's quite a thick membrane, a bit different than the cell membrane. Okay, this is what the nucleus looks like under a scanning electron microscope. Okay, it's this picture right here. It gives you an idea. That's 1 micrometer there. So your nucleus might be 2, 3, 4, 6 or 7, 6 to 10 micrometers across. Okay, it's kind of a range for how big they would be. So you can see them within a cell, but they're not huge. Okay, they're the biggest thing within the cell usually, but so that's kind of what they look like. If you zoom in even more, these are what the nuclear pores look like. Okay, they're about a tenth to a quarter, okay, of a micrometer in size. So it's not something you would likely see with our microscopes. Right? And if you zoom in even more, you can actually see the kind of intercrossed fibers that make up the membrane of the nucleus. Okay. All right, questions on the nucleus and the nuclear membrane. So what does the nucleus do? Stores DNA, okay? If I asked you on a quiz, identify and give the function, and one of the things was the nucleus, you'd say nucleus stores DNA. That would be sufficient, okay? Now, protein synthesis is also controlled from the nucleus, but it is done specifically by the nucleolus. Okay, so it is the nucleolus, which is inside the nucleus and is part of the nucleus, that actually controls the protein synthesis. Right? It's that librarian that goes around and finds the book you need and makes a copy of it for you and lets you have it. Okay. All right, so if I labeled the nucleolus on a diagram and asked you to tell me what it did, you would say controls protein synthesis. So this is how that happens, okay, how the process of protein synthesis occurs. Right? Within the nucleus, so the nucleolus produces RNA, which is a single strand of DNA. Right? If you recall, I think you learned this in Science 9, but DNA is 2 strands, but the strands are complementary. Right? A always goes with T and C always goes with G and so on. So you really only need to know one side because you know what the other side would be. All right? So that's what the nucleolus does, is it essentially unzips the DNA, okay, so pulls it apart like this, and then copies the section of the DNA that it needs, makes that single strand. So that's what this pink line is right here. Okay, so this pink thing here, that's the copied DNA or RNA. That goes out through the nuclear pores, so you can see that happening right here, okay, and that RNA then goes to a ribosome. Okay, so that's what this little brown thing is. Okay, and the ribosome begins to read it. Okay, the ribosome reads it and starts collecting raw materials. That's these purple shapes here that are all throughout the cell. Okay, it starts collecting those materials from within the cytoplasm inside the cell, and it starts putting them together in the proper order. So these little raw materials are really amino acids. That's what you use to make up a protein. Okay, it puts them together in a certain order. That makes a certain kind of protein, and that's how protein synthesis works. Okay, so the nucleolus copies the DNA. The ribosome reads the DNA and builds the protein. Okay, kind of a three. It's more complex than that, but that's the level you need to know it at. Okay, the biochemistry of it isn't important right now. All right. Okay. Yeah, and obviously this doesn't happen within the nucleus, okay, for a couple of reasons. We don't want raw materials always coming into the nucleus because they could be bringing in bad things, and there's a big membrane there. It would really restrict the flow of that raw material, so it's better to do it in the cytoplasm. Okay, inside the cell, if there's one organelle that takes up more space in the cell than any other, it's the ER. Okay, there are 2 kinds, rough and smooth. They are very, very important. Smooth, probably more important than rough. All right, but essentially they can do similar things. So within the membranes of the smooth ER, a bunch of things go on. Okay, your cell membrane is a wall, and it protects the cell from lots of things. But if your cell is constantly under attack, the cell membrane gets damaged. Okay, things are trying to get in, but they don't. You twist it, whatever, you damage part of it, it stretches, it damages part of it. The membrane always has to get repaired. So one of the jobs of the smooth ER, okay, is to make lipids. Okay, because lipids are what membranes are made out of. Okay, fats. That's what your membranes are made out of. All right, the reason they are made out of that is because fats and water don't like each other. So as a result, water gets trapped within the cell because it can't get past the fatty layer that's on the outside. All right, now there are ways for water to move across that membrane, but for the most part, it stays in there. Okay, it's what we call a hydrophobic or water-fearing barrier, okay, that keeps water in or other materials that are dissolved out. Okay, so it's an important thing that the smooth ER can synthesize different kinds of lipids and make these membranes. This is why, guys, fats are an important part of our diet. Okay, now, we as North Americans eat way too much. Okay, but we still need fat in our diet. It's important to get fat in your diet. If you were to eat nothing but vegetables and fruit for your whole life, it would actually be somewhat unhealthy because you would not get the fats that you need. Okay, which is why people who are vegan typically have to supplement their diet with like nuts and things like that that would contain proteins and fats, because we need those as the building blocks for things that go on within our cells. You cannot live strictly on a, let's say, vegetable and fruit-based diet. You have to have other things in there. If you, I mean, you can live without eating meat, but you can't live without eating proteins and fats. You still have to get those somehow. All right. You can get those in things like coconut oil, right? Olive oil, things like that. They're plant-based, okay, but they're still fats, and we need those. All right. Does that sort of make sense? So, I mean, if you're thinking about, oh, I gotta limit how much fat I intake. I mean, yes, we as North Americans in general could eat a lot less fat, okay, but we still need some. Okay, it's still healthy to have some. All right, complex carbohydrates can get broken down within the smooth ER as well, and that's important. The lysosome, its job is more to break down really complex things, not so much carbohydrates, but like proteins and fats and things like that. That's what its enzymes are designed for. Okay, the smooth ER is really designed to break down really long sugars. That's what complex carbohydrates are. So fructose would be an example. Sucrose would be an example. Those are complex carbohydrates. Starch is a complex carbohydrate. Right? Those things get broken down into glucose by the smooth ER. Right? So that's a second job. Okay, breaks down complex carbohydrates. Okay, and detoxifies drugs and poisons that might get in there. Okay, also a very important job. Our bodies naturally produce some things that are toxic. Okay, if you are engaged in incredibly intense anaerobic exercise, your body will undergo lactic acid fermentation. You guys have all felt the burn before, right? Okay, in your muscles, you know, you're working real hard and you get that burning sensation in your muscles. That's lactic acid. It's toxic. That's why it burns. Okay, and your body has to have ways to get rid of that. One of the places where it can be broken down into less harmful materials is the smooth ER. That process, however, requires oxygen, so it doesn't happen until you slow down and replenish your oxygen debt. Okay, that sort of makes sense on that? Okay, that's why if you stop and breathe heavily for a little while, the burning sensation goes away. Okay, hormones can also get produced in the smooth ER as well as the rough ER. Okay, it can be produced in both. Okay, those are important, and they can be quickly then sent to the cell membrane because the ER is usually in contact or touching the cell membrane in various places. Okay. All right, so the smooth ER has lots of jobs. My expectation would be that if I asked you what does the smooth ER do, that you could tell me at least a couple of them. Okay, I wouldn't expect to get all 5. Okay, but I would expect that you would give me at least a couple. Right, probably on a test 1 or 2, okay, would be sufficient. You demonstrate to me that you remember what it does. Okay, the primary jobs would be lipids and detoxification. All right, rough ER, we didn't talk about it so much here, but the rough ER mostly transports protein. Okay, so I just want to write. Okay, and that's because it's covered in ribosomes, which are busy making that stuff. Right, and so it's able to quickly move it around from place to place. That's also why you usually see it attached to the nucleus. Because where's the information for building all that stuff coming from? From the nucleus. So it makes sense to have them close together. So when we do our cell anatomy lab on Tuesday when we get the lab, okay, we will be looking at animal cells, plant cells, etc. And sometimes you can see the rough ER. Okay, it's usually you can see it near the nucleus. It almost looks like the nucleus has a beard. Okay, so if the nucleus is round, there'll be this kind of like almost slightly triangular, blurry thing kind of attached to it, and that's the rough ER. That's where you can look for that. Okay, if it looks like your nucleus has a beard, okay, you're looking at the rough ER. Questions on the ER? All right, and remember, you can just write ER, okay, on a quiz or a test. Understand that it stands for endoplasmic reticulum, that on a multiple choice question, I would write it as endoplasmic reticulum, but you guys can save yourselves time in writing, okay, and just abbreviate it as ER. All right, lipids can be moved quickly to the needed sites, and poisons and drugs can be detoxified and quickly removed from the cell through the cell membrane. And as long as all that stuff stays in the ER, it doesn't touch any other parts of the cell where it could cause damage. Okay, so that's again the the benefit of having all these separate compartments within the cell, is you can really localize where things end up. So under an electron microscope, this is what rough and smooth ER looks like. Rough ER is very layered, okay, kind of very structured, and you can see that the ribosomes are kind of stuck all over it, all these little dots here, but it's very kind of linear. Smooth ER is just folded over and wrapped and spiraled around, okay, kind of all over the place. It's much more random looking, right, and obviously doesn't have ribosomes all over it, so by comparison looks smooth. All right, Golgi apparatus. Okay, some of the stuff that's in the ER, especially if it's a detoxified poison, will make its way to either the membrane or the Golgi apparatus. And from the Golgi apparatus, it will definitely be on its way out of the cell, because the Golgi apparatus is really just a garbage man. Okay, it's going to package the waste, possibly process it a little bit more, and then send it toward the membrane in what we call a transport vesicle, okay, which is basically a bubble made out of the same material as the membrane. So that stuff will just find its way to the membrane. Okay, when it hits the membrane, it kind of merges with the membrane and just pops out. All right. Okay, so the Golgi apparatus basically looks the same in every cell. It is something that you can sometimes see in our cell anatomy lab if you get a particularly good animal cell or particularly good plant cell. You can sometimes see it. It does have a pretty distinctive shape. Right? It'll look like kind of a dark set of lines that tend to curve a little bit, kind of down, almost like a frown. Okay, it'll kind of look like that, and sometimes you'll see it. Usually, though, okay, usually you won't, but you can sometimes see it. But when you look at it under an electron microscope, you can really see that one side of it is busy sending stuff away. You can see all these big vesicles here. Okay, each one of these things is a transport vesicle. But on this side, everything is very, very small. Okay, because none of this stuff has been packaged yet. Okay, hasn't been packaged or processed by the Golgi yet. So it comes into the Golgi packages it and sends it out in these vesicles, which are bound for the membrane, and then they'll be out of there. Right, so there is a cis face, okay, that's this side, the side that receives everything, and a trans face, the side that ships everything and gets rid of it. Okay, you won't have to know cis and trans. Okay, just know that it packages stuff and sends it to the membrane. All right, so the Golgi apparatus, okay, essentially transports material to the outside of the cell. Okay, questions on the Golgi? All right, lysosomes. So we've talked about lysosomes already. We're only going to find them in what kind of cell? Okay, animal cells or the cells of any organism that is a chemoheterotroph, okay, because we could find them in a fungus cell, right, and not just in an animal cell. So anything that's a chemoheterotroph is going to have these, okay. They have really strong membranes because the stuff that's inside of them would eat the cell. That's what it's designed to do, because what you eat is cells. You're eating the tissue of other organisms that contain cells. If you're going to break them down for food, you have to have enzymes that are capable of breaking down cellular material. So it's really important that the membranes for these are very, very strong so that those toxic materials don't get out and eat the cell. All right? Now, sometimes, okay, these little vesicles will come from the Golgi apparatus and they actually become the lysosomes. The Golgi is what makes lysosomes. Right, so it makes the enzymes that can eat the cell. Okay, those will merge with what's called a food vacuole, so a particle of food. Okay, and they will digest it. Okay, now the cell will also engage in what's called autophagy, which means it's recycling organelles that have ceased to function. Right, so like the equivalent of saying, "Oh, I broke my hand, so I'll just eat it." Weird, but that's what cells do. Okay, they're kind of the ultimate recycler. Okay, there's no sense in getting rid of all that raw material. They could digest it, and the cell could build a new mitochondria from there. Okay, if the organelles were damaged or no longer functioning. Okay, so obviously very important that they have a thick membrane. So lysosomes do what? Break down nutrients. I'd be satisfied with digest food. Okay, if you were writing down something like that on a test. The other job of a lysosome, and this is a pretty rare occurrence, okay, but they can be responsible for what's called cell lysis. Okay? A cell will sometimes kill itself. If the cell detects that it is not functioning properly and is a danger to surrounding cells as a result, it'll take one for the team. Okay? And it will rupture all the lysosomes within the cell, and the cell will eat itself to death. Right? That is an important mechanism within cells. When we're young, that mechanism functions very, very well for the most part. As you age, the cell's ability to detect malfunctions decreases. Okay? The more times a cell divides, the less able it is to detect malfunctions, which is why cancer is more prevalent in older people. Right? The cells are unable to detect that they are doing something wrong. They begin dividing out of control. They don't sense anything is wrong, and they begin to essentially destroy the surrounding cells, and they become a tumor. Okay, that's essentially how cancer works. Okay, cancer is your own cells dividing out of control and using up all of the nutrients that the healthy cells would have used, and it essentially kills off the healthy cells and becomes a mass of malignant, non-functioning cells. All right, does everyone kind of follow me there? Okay, so that's why you get like people often say, oh, no, there's a lump here, or and kind of got this weird sensation in this area. Okay, that's the lump of cells. Okay, that has just divided out of control, and that's your tumor. All right. The problem with cancer is your body doesn't know that there's anything wrong because those cells are all your cells. Okay, so it's going, well, there's nothing foreign there. Those cells are us. Those are our cells. They look like us, so we'll just leave them alone. And that's the problem with cancer, is it's disguised from your own body because it's your own cells. Okay, questions there? Okay. All right, the mitochondria. Okay, mitochondria are kind of a sausage-shaped organelle, and they have a double membrane. Okay, not a single membrane, but a double membrane. And there's kind of a space between the inner membrane and the outer membrane that acts as an insulator. Because the mitochondria are busy breaking down sugars and releasing their energy. Okay, and when you break down sugars to release energy, remember we looked at this yesterday. Oh, that 6 is way too big. Hey, if you recall us talking about that yesterday, that's what kind of a reaction essentially. It's a combustion reaction, which means it's going to produce some heat. Okay, so the mitochondria will produce some heat while they're carrying out cellular respiration. Okay, the breakdown of glucose into carbon dioxide and water. Right, that heat could be detrimental to the functioning of the cell. Your cells are all designed to function properly at about 37 degrees Celsius. Okay, if you run a fever, then these cells or these organelles, sorry, are burning more sugar than they're supposed to. The cell begins to run hotter than normal. Okay? If all of your cells are doing that, your temperature goes up. Okay? And usually your body also shuts down the cooling system for your body, which is sweating. Okay? If you have a bad fever, you usually don't sweat very much while you're getting hotter. Okay? And when the fever breaks, then you just get drenched in sweat because your body's trying to cool itself off. Right? What's the purpose of running a fever? Why does your body do that? Okay. Well, your body normally functions at 37 degrees. Any organisms that get in your body to try to make you sick probably like that temperature too. If you raise the temperature of the human body, the processes, chemical reactions, and things like that that the body uses to function don't work as well. Okay? The enzymes that help things to work are designed to work at 37 degrees. If you're hotter than that, those proteins break down, and then the reactions they're supposed to help go on don't go on. Okay, which is why you just feel sapped when you have a fever, right? Because you can't carry on a lot of the stuff that needs to go on. If your body gets to a certain temperature, you could die, okay? Because the functions and processes within your cells just break down and your body just can't run, okay? It just can't run at that temperature, essentially melts down. Not literally, but essentially. And the same thing is true for hypothermia. If you get too cold, same thing. The proteins that help the reactions within your body don't work, the reactions don't go, and you just get sleepy and that's it. Okay, everyone follow me on that one? Okay, so the mitochondria are responsible for getting all that energy going, but they have to insulate the cell against the rest of, insulate themselves against the rest of the cell, or vice versa, insulate the rest of the cell against themselves, okay, because they do produce a fair amount of heat, so they got a thick double membrane there. All right, so mitochondria essentially burn sugar for energy. If you wanted a simple function, they burn sugar for energy. Okay, or you could say carry out cellular respiration if you wanted, that would also be true. All right, chloroplasts, only going to be in plant cells. Okay, they also have a double membrane, right? They absorb a lot of solar energy, so as a result, they can also get hot. Okay, but they also want to make sure that all the pigments that are part of photosynthesis, chlorophyll being the main one, but there's other ones as well, that they're all stored, compressed, and whatever in one place. Okay, in these organelles. These organelles can be moved around within the cell. Okay, why would that be important? Why would the cell want to be able to move them around? Okay, see this plant? Does one side of it get more light than another? Okay, so you probably want your chloroplasts to be on the sunny side of your cell. All right, so they do have, the cell does have the ability to move them around a little bit and put them on the side of the cell where they're going to receive more sunshine. Now, you're probably thinking picotear cells are really small. Does it really make that much difference? No, not really, but it still has the ability to do that to some extent. All right, so we got the stacks of thylakoids inside. Okay, you probably won't see that, but you can see chloroplasts when we look at our plant cell in the cell anatomy lab on Tuesday. Okay, there'll be green dots, okay, and they'll be reasonably large. Right, they're not quite as big as the nucleus, but you can see them, and they're usually near the edges of the cell. They usually get pushed against the wall. All right, so you can definitely see those. All right, so main job, chloroplasts. What do? Carry out what process? Photosynthesis. That's what, if, on a test or a quiz, chloroplasts carry out photosynthesis. Okay. Right, and one of the first things you'll learn in Bio 20 is how photosynthesis works. Okay, the first unit of Bio 20 is like biochemistry. It's cellular respiration and photosynthesis, and they're way more complex than you thought they were. Right? Yeah, there's lots of enzymes and big things that go on in there. Okay, other organelles and functions. Okay, so the cell membrane. Okay, just kind of blow this picture up here. No, except it's behind other things. We'll move it over here. Okay, so the cell membrane isn't as simple as you might have thought it was. Okay, it's actually got a lot of stuff in it. Okay? It's kind of like a matrix of many things. It's if you've ever made Jell-O with fruit in it, okay, and all kinds of, if you put all kinds of things in the Jell-O before it sets, that's what a cell membrane is really like. There's the fatty layer that's the majority of the cell membrane, but then there's all kinds of other things. There's special kinds of proteins that are embedded with sugars called glycoproteins. Okay, there's globular proteins, which are just really big ones. There's transport proteins that help move large molecules through the membrane. They're almost like gates, okay, that can change their shape and allow certain molecules to get through. Right? There's all kinds of additional structure to the membrane. You'll never see any of that, okay, because it's incredibly small. At least not with our microscopes, we wouldn't see that, right? But if we're looking at the membrane, all we're going to see is just a line that kind of tells us that's the cell and the rest of it isn't the cell, okay? But this is all the stuff that's actually in there, so it is a lot more complex than we initially thought. There's also receptors all over the surface of the membrane, okay? Receptors are like antennas. Now, if you're driving along in your car and you're listening to the radio, you have one antenna, and it can pick up all the stations. But cell membranes don't work that way. They have to have an antenna for every signal they could receive, because their signals are chemical. All right, and so in order for them to know that a chemical signal has been received, they have to have a receiver that is the same shape as the signal. All right, so it's like puzzle pieces. You can only put this puzzle piece in that puzzle piece. Okay, they fit together because they have the same shape. Everyone okay with that? Okay, you can't put like a, I don't know what, you can't put a three-pronged plug-in into a two-pronged outlet. Okay, they don't fit with each other, so they have a receptor for every chemical signal that they could possibly receive. All right, that out of the way. Okay, so the cell membrane, and this is what you would write on a test if you were telling me what the cell membrane does. Okay, it controls endocytosis and exocytosis. Okay, remember we said cyto means cell, okay, and tosis is transport. So exocytosis is transport out of the cell. Endocytosis is transport into the cell. All right, that's what the cell membrane does. That is its primary function. Okay, so that is what we would write. All right, so if you write controls what gets in and out, I'm going to go not good enough. Okay, I want the technical terms for getting in and out of the cell, and that's endocytosis and exocytosis. Now, the cell wall. Remember I told you the other day that it's not a thick, solid wall, it's more like a frame, okay, if you recall that. So the cell wall, at least of plant cells, is made of cellulose fiber, and it's a frame. It keeps the shape of the cell. That you'd write something like that for the function of the cell membrane. Keeps the shape or supports the cell, and this is what the cell wall looks like. Okay, the green part there is the cell wall of a plant cell. It's hollow. Okay, it's got holes in it. Right? That's what Hooke could see when he looked at cork, because it stays there even after the cell dies. Right? The cellulose doesn't break down very quickly. That's why we use wood to build houses. Wood is made out of cellulose. Okay, paper is made out of cellulose. So it's a kind of a long-lasting fiber, and you can see, like I said, that it's hollow. Okay? Anything can pass right through it, but it's strong. It's a good frame. All right, water vacuole. Water vacuole is just a big balloon full of water, okay, that's going to be in the middle of the cell, and it's going to, as it fills up, push everything else out of the way and against the cell wall. All right? In doing that, it creates pressure from within, okay, that can support the cell. Right? It would be kind of like an airbag, except it's using water. You know, if your airbag inflates in a car, it can support the pressure of your head hitting it, and then it'll slowly and then kind of push back. Okay, if the airbag didn't inflate or was empty, you would just go straight forward, and there would be nothing there to support you. Okay, so it's pushing from the inside and supporting from the inside as a result. Okay. Oh, sorry, better example. Better example. If you've ever had a bottle of fluid, let's say water, and you drank a little bit of it and then you put it in the fridge or in the freezer, you ever notice that the bottle kind of goes, it kind of sucks in a little bit, okay, when the temperature decreases? Because then it's not exerting as much pressure on the container, and so the atmosphere can actually crush it a little bit, okay, because there isn't enough pressure from within to support the shape. Okay, it doesn't take up as much space. Well, that's the same thing with the water vacuole. Pressure from within pushes out on the cell. If there's no pressure in there, the environment pushes in on the cell, and that's when the plant can wilt and look sick. Okay, centrioles. Talked about these. They're only in what? Animal cells. And the water vacuole is only in plant cells. Okay, so centrioles, okay, are these structures that help with cell division. And I was talking about these cables and all that kind of stuff, okay, yesterday when I was describing what they do. All right, so you got two centrioles, and this is kind of what they look like under electron microscope. Okay, this is how they kind of reproduce. And then when they go in during cell division, they send out these little cables that attach to your DNA. Okay, so these are your chromosomes here, right? They send a cable that attaches to them, and then when the cell divides, it pulls those things apart. Okay, some of them go this way, some of them go this way, and that makes sure that each new daughter cell is what they're called, okay, ends up with all the DNA that it requires, right? So centrioles, really, all you need to write is helps with cell division or pulls DNA to one side or something like that, okay, would be sufficient for kind of a quick answer on those. Okay, and lastly, microfibers and microfilaments. Okay, we also labeled microtubules on our diagrams yesterday. They all do the same thing. They're part of the cytoskeleton, okay, and they help the cell stay intact during changes in shape, okay. You might just want to add to that, okay, that we talked about yesterday, cytoskeleton. Okay, they're part of the cytoskeleton. Okay. Okay, so the next topic we got to go over, now that we know what all the parts do, is we got to talk about how the cell moves material. Okay, now, moving material, okay, if we're trying to move stuff within the cell or into the cell and out of the cell, we want to avoid using energy if at all possible. Okay, because the cell only has a finite amount of energy and it needs that to do things. Okay, it doesn't want to be spending energy on something if it doesn't have to. So the cell tries to use as much as possible what we call passive processes. Okay, so a passive process, okay, uses no energy. Okay, an active process uses energy. Okay, so a passive process is essentially free, and an active process costs. Right? Now, I think we'd all admit that if we could get everything for free, we'd be pretty happy about that. There are some things that we can get for free, right? You know, like, you know, you go on the App Store and, oh, that app's free. Cool, I'll get that. Oh, there's a paid version. Nah, I'm not going to pay for that. I get the free version. What do you usually notice about the free version? Yeah, they suck. Yeah, okay. I mean, they sort of do what the paid version would do, but they don't do it nearly as well, right? There's something, they're either slower or they don't, you know, there's a feature that they're missing or they don't have the ability to do something that the $12.99 version would do, okay? So you get what you pay for in life and in cellular transport, okay? You get what you pay for. A passive process will do the job. It will often do it fairly well, but there will be certain things about it that are not ideal. All right, so that's what we're going to look at here. Okay, the first cellular process that we're going to look at is the one that is used most often. The most common way for any living organism to move something from place to place is by diffusion. Okay? And diffusion is just the natural tendency of any material to move from an area where it is very concentrated to where it is less concentrated. All right, so it would go from, it would be kind of like if you were in a really crowded room where you were just, you know, packed in really tight, and you probably wouldn't like that, and if everyone's in your personal space, your desire is that you want to get out of there and have more room, okay? Well, that's a natural tendency of all particles, be it in air, in water, in a cell, whatever. The natural tendency of things is to move from an area of high concentration to low concentration. That is the process of diffusion, okay? It's how a fart works. Okay, if somebody farts in one part of the room, okay, and you just happen to be the unfortunate soul who is right next to them, okay, it's a lot worse for you because right there it's very concentrated. Okay, if you're on the other side of the room, by the time it gets there, it's kind of dissipated, right? I mean, you detect it, you're like, that's unpleasant, but I'm not going to gag. Okay, and it just spreads out and kind of goes everywhere, okay, over the course of time. Everyone follow me there? Always from high concentration to low concentration. Okay. All right, so molecules have what we call an intrinsic kinetic energy. That's that they're always moving. Okay, that's the particle theory. You probably heard about that at some point. Molecules are always moving except at what temperature? It's very cold. Which zero? Not 0 degrees Celsius, but absolute zero. Okay, at absolute zero, all that intrinsic kinetic energy is gone. They don't move at all. Okay, but in any other case, there's this natural movement, and that natural movement will cause them to move away from each other because they run into each other, they bounce off, and they kind of spread out. Okay, so that's thermal motion or heat. Okay, and one result of that motion is diffusion. It's a tendency of molecules of any substance to spread out into the available space, okay? Or move from high to low. Okay, so it'll move from where it is more concentrated to an area where it is less concentrated, or we sometimes call that down its concentration gradient. Okay, and that's what you can see in that picture there. Dye is being kind of injected into this water, and you can see that it naturally spreads out. It's very concentrated and very dark here where it's coming in, okay, but it spreads out and becomes more and more faint, okay, as it moves away from that area of high concentration. Right? If you ever put a drop of food coloring into water and watched it, you can see that it kind of randomly kind of makes little swirls and stuff, but very quickly dissipates from that really dark single drop to being a fainter version of itself where the whole entire jar or glass or whatever is now that color. Okay, that's diffusion, that's that process. So your cells naturally use that process to move things. Now, kind of makes sense for them to use it. I mean, yes, it's free, but if it needs something in one place, it's probably because it's very low concentration in that place. So things are naturally, by diffusion, going to move to where they're needed. Agreed? Right? They're going to be produced here. Well, if they're being produced here, there's going to be lots of them. Okay, it's going to be very concentrated here. But over here is where it's needed. If it's needed there, it's because there isn't any there. So diffusion will naturally move stuff in that direction. Is it targeted, though? No, there's no guarantee all of it's going to get there. It's not. It's going to naturally move in that direction, but not all of it will get there. That's the drawback. Okay? Diffusion is the shotgun approach to delivering material. Okay? It's not great. All right? It'll get the stuff there, but it'll get stuff a lot of other places too. Maybe not always where it's needed. Okay? If you had a truck, you could transport it from here to there, and you would know that all of it would stay in the truck and it would all get there. But if you're doing that, that's not passive. It costs. Kind of get the idea here? So diffusion is used most often because it's free and it's reasonably reliable, and over short distances works pretty well. Is a cell going to be a short distance? Yeah, okay, cells aren't very big. So realistically, if you're making something and it only has to diffuse from one part of a cell to another, it's going to do a pretty good job of getting it there because it doesn't have time for it to really spread out. What if a cell gets really big? Will that work as well in a really big cell? No, okay? That's kind of why cells don't get really big. Okay? Diffusion kind of limits the upper size of a cell. If they get too big, diffusion won't work. Okay? And then they'll spend energy transporting, and they just can't afford to do that. Okay? Everyone, is that making sense to everybody? Okay. So diffusion, most common. Okay? Most common transport. Okay, and it's passive. All right, osmosis, next type of cell transport. Now, osmosis is a specific type of passive transport. So it still doesn't cost anything, doesn't cost any energy. But osmosis only transports one material, water. Okay, osmosis only transports water. Okay, and osmosis, very simply, is the movement of water across the membrane. Okay, now we said the membrane's made of that fatty stuff. It's hydrophobic. For the most part, it kind of keeps water in one place or the other. But if there's a real difference in solute concentration, so salts, okay, on one side of the cell versus the other side of the cell, water will move towards the salt. Okay, because it wants water naturally, and membranes naturally want to have equal concentrations of things on both sides. They want a balance. Okay, so water will always move towards the salt, okay, and try to balance the concentration on both sides of the membrane. That's what osmosis is. Okay, it's a passive movement of water across a membrane to balance salts. Okay, so that's kind of the definition right there. Now, I use the term hypertonic and hypotonic in there. Hypertonic means lots and lots of salts. Hypotonic means very few or none. Okay. So what you'd see, and I think you do this lab in Bio 20, okay, you take a U-shaped glass tube and you put a piece of sausage casing across the middle of it. Okay? Sausage casing is made of the same material that you are made out of. It's made out of cells. It's the intestinal lining from pigs or whatever. Okay, on one side of it you put like distilled water, and on the other side you put salty water. And if you leave it over the course of time, and not very much time, the water level on the side that's just water drops, because water is moving across the sausage casing and over here to the salty water to try and make the concentrations on both sides the same. Okay? Now, it's a passive process. That means your cell can't control it. It just happens. Okay? So it's good in that it's free, but when it's free, you can't control what it does either. That can be bad for a cell. What happens if you drink salt water? You get really dehydrated. Okay? Because if you consume salt water, your body puts that salt into your blood, because that's the only way to get the salt to your kidneys where it can get rid of it. But while it's in your blood, it's drawing water out of your cells because your blood gets really salty. Okay? And so your cells, they can't control where the water goes. It's just the water goes, hey, it's really salty over there. Let's go. Okay? And across the membrane, all the water goes into your blood. Okay? And you start urinating out all this water. Okay? Because then your kidneys suddenly detect, wow, the blood's like really watery. It's got tons of fluid. We got extra fluid in the body. We've got to get rid of it. Okay? And you just start urinating all of it away. Okay? Well, you're not urinating the salt water away. I mean, you are, sort of, but you're also urinating away all of the water that was in your tissue. So you actually dehydrate way faster. Okay? So if you're ever stranded on the ocean in a boat, don't drink the water. Okay? You'll die of thirst faster, die of dehydration faster by drinking the salt water than you would by drinking nothing. Okay? In fact, for the first day anyway or two, you're actually better off to drink your own urine than you are the salt water. That's disgusting, but it's true. Okay? There would be less salt in your own urine than there would be in the ocean water. Okay, so don't do that. Just don't get stranded on the ocean. Okay, but don't drink salt water either, because it's not good for you. All right, it'll draw all the fluid out of your body. That's often why you have that sensation, and maybe you've had like a big thing of fries or something really salty, and an hour later you're like, man, am I ever thirsty? Okay, well, it's because you just ate all this salt and your body's got it in there and it's drawing water out of your tissues, and your body goes, hey man, we're getting dehydrated, we got to drink. Okay, make that guy feel thirsty, okay, and then you'll drink. Right? That's kind of how all that works. So all of that is caused by osmosis. It's also the reason why if you put salt on a slug, the slug shrivels up. Okay, all right. If you pour salt, because they don't have skin like we do, it's not a kind of impermeable barrier like ours. You pour salt on them, and water just comes rushing out of the slug to get rid of the salt, and the slug just shrivels right up really fast too. Okay, so yeah, don't do that. It's cruel, but that's why it happens. All right, so does that sort of make sense to everybody? Now, the opposite is true. Your cells always contain some salt. If you become, let's say, overhydrated with fresh water, your cells will start to absorb the water out of your bloodstream, because the water will just move naturally into your cells out of your blood, because there's more salt in your cells than there is in the blood. Okay, that's what we want. That's the situation that helps us to absorb water into our bloodstream and into our cells. That's kind of the process that naturally goes on. Okay, however, if I was to put a normal cell into distilled water, it would pop, because it could never balance the concentration. Distilled water has no salts in it, none. So if I put it in there, water just moves into the cell by osmosis. The cell can't stop it because there's salts in the cell, and the salts can't get out. Okay, so water just keeps pouring into the cell until the cell just pop. Okay, bursts, and then the concentrations are balanced after that, because the cell is popped. Okay. All right, so does that sort of make sense? It's a passive movement of water. The cell can't control it, but for the most part, it gets things done, and it's free. So the cell continues to use it. All right, so the big things to remember about osmosis, okay? Passive transport of water only, okay? And it's across the membrane, okay? And we're trying to balance salts. Okay, when we say salts, plural, many different kinds, not just sodium chloride. Okay, there's lots of different kinds of salts in your body. Okay, so we're going to cover the big three. Maybe we'll get through the big three today. Okay, the big three are diffusion, osmosis, and active transport. Okay, so diffusion and osmosis were passive. Active transport, active. Active transport requires energy from the cell. Okay, so that's the big thing. Okay, active transport is, in fact, active, and it is the pumping of solutes against their gradient. Okay, that means the opposite direction, from low to high. Okay, it's pumping water uphill. All right, water naturally passively runs downhill, but if you want to move it uphill, you have to pump it, you have to use energy. That's what active transport is doing. Okay, your cell is saying, I want to hoard a whole bunch of this stuff. Okay, I got a bunch of calcium in here, but I want more. Pump it in here. Okay, because it's not going to move in here on its own anymore. Okay, it's looking in the door and seeing that it's crowded in here and there's lots of calcium, but we want more anyway. Okay, and it's pumping it in. It uses energy, and it uses transport or carrier proteins on the membrane to do it. Okay, changing the shape of those proteins requires energy, and really all they do is open and close like this. Okay, and in the process, they can move these solutes across the membrane. Okay, that's active transport, just the movement of solutes across the membrane from low to high concentration. So you can see out here, we got lower concentration of ions inside the cell, higher concentration of ions. Right, so active transport moves things from low to high and uses energy to do it. Okay. Right, so uses energy. All right. Don't forget your microscopy labs due tomorrow. If you want me to look those over, scheduled help or lunch today.